So this is my favorite brain part presentation for Dr. B's psychobiology class, and my name is Megan Antle, and let's get started. So I chose the dorsolateral prefrontal cortex, or DLPFC is the term I'm going to be using during this presentation so I don't get tongue-tied. So this is just a brief table of contents over what I'm going to be discussing. We're going to go through an introduction, functionality, damage, the relationship between OCD and the DLPFC, and then a quick conclusion and a resources page. So this is the structure and anatomy of the DLPFC. In figure 1, you're able to see the location of the DLPFC within the brain. And in figure 2, you're able to see a study done by Sala and colleagues, which included a parcellation scheme, which is what figure 2 is representative of. The 3 clusters that you can see in red, blue, and green are representative of subregions of the DLPFC based on this parcellation scheme. So the functionality and purpose of the DLPFC is why it's my favorite. It's traditionally associated with general executive control functions or higher cognitive functions. So it's very important. It controls functions like task switching and reconfiguration. It prevents interference, inhibition, planning, working memory, and maintaining of abstract rules. The DLPFC has also more recently been found by Hertrich and colleagues to be activated in speech and language tasks. However, further research will need to be done on this language role for more detailed descriptions of its functioning. Recent research has also found that reward influences activity in the DLPFC. It's been reported by Kobayashi in the Encyclopedia of Neuroscience that when animals are presented with different reward outcomes, the DLPFC neurons show sensitivity to both what the animal must try to remember and to what they will be rewarded with for performing the task. The combination of representative memory contents and reward outcome tells researchers that the expectation of a reward has modulatory effects in the DLPFC's working memory process. So the DLPFC sends afferent projections to the dorsal striatum, and this plays a role in damage of the DLPFC. Structural imaging has shown researchers that cocaine users have a significant reduction in cortical thickness in the DLPFC. In a PET study done, abstinent cocaine users were found to have less activation in the DLPFC when participating in a reward-related or decision-making tasks compared to non-users of cocaine. Less bold signals in the DLPFC during an attention task in cocaine users before entering treatment predicted longer stays in treatment. So this is indicative of that when decreased activity in the DLPFC during this attention task could possibly be linked with more efficient processing and greater decision-making abilities. Because the DLPFC is considered the central executive, those with damage to the area may experience cognitive difficulties in memory retrieval, strategy generation, behavioral response generation and organization, and internal cues utilization to guide behavior. This is also similar to executive dysfunction syndrome. So obsessive compulsive disorder, or OCD as I'm going to be referring to it, does interact with the DLPFC. In a study done by Vahabzadeh and McDougall, which is supported by multiple previous research studies, it was found that volumes of DLPFC are reduced in persons with OCD. One study found that increased OCD severity was correlated with decreased DLPFC volume. PET neuroimaging has shown that the DLPFC is hyperactive in persons with OCD. The DLPFC abnormalities are very likely associated with a deficit in executive functioning, which is also seen in OCD. The non-pathological cognitive processes are or can be impaired because the inability to appropriately allocate resources could lead to over-allocation of cognitive attention to obsessive thoughts. So, the dorsolateral prefrontal cortex, or DLPFC, is an executive function. When it is damaged, the results are severe in working memory and selective attention, or in other words, it is very important, which is why it is my favorite brain part. And these are my resources.